Hi everyone, hope my slides are visible and you can hear me. Sorry about starting the class a bit late. So today we will be covering cell segmentation. Specifically, we will look at a software called Cellpose to do that. So what exactly is segmentation? So segmentation means you have an original image. For example, here this looks like a image of main nucleus, and segmentation means identifying each particle as separately. So here, when you look at it with human eyes, you know that this is one nucleus, this is one nucleus, this is one nucleus, and so on. So that is similar to doing manual segmentation, like we were drawing the outlines of cells in the previous classes. But there are many microscopy images where there are many cells in the same image. Also, you might have many different images, so it may not be possible to sit and do it manually. So that's why there are softwares that can automatically segment the cells or nucleus or whatever is in your image, and it gives you the cell outlines and output mask like this. So we'll talk more about what exactly this output represents and how is this saved and so on in the upcoming slides. So there are like two types of segmentation broadly. So one is semantic segmentation. In this, it just identifies like background versus particle, but it may not know that this is a different particle, this is a different particle, etc. But in instant segmentation, it differentiates the particles from the background, and it also differentiates between each particle. So you know that this orange is a separate cell, green is a separate cell, pink is a different cell, and so on. So if you just want to do something like you want to get the background which without any particles, then this first approach may be useful. It is also simpler. But if you want to characterize, say, the shape of the cells and so on, which is typically in biological images, that's what we want. So in that case, instant segmentation is more helpful. So one of the softwares that has been recently developed to segment cells automatically is called Cellpose. So first version of Cellpose was it was out in 2020, and there is also a paper published on it. So if you want to go more into the technical details of how it works, the um, exact AI model that they trained on and so on, then you can refer to these paper. So in the first version of Cellpose, it was just working on the models that they had already trained. Um, but in the second version of Cellpose, the good thing is that you can also train your own model for the data set that you have. So, for example, if you have very elongated cells, then the Cellpose model that is available publicly that they had trained may not be able to segment it properly. So in those cases, you can also train your own model with Cellpose 3, Cellpose 2. And then very recently, in this year only, Cellpose 3 was also out, which in which they had trained a better model. So like there is Cyto, Cyto 2, Cyto 3 model like that. We'll see in the upcoming slides. And one more improvement with Cellpose 3 is that it also has more advanced image processing so that the cell outlines are more clearly visible and segmentation is also better. So essentially, Cellpose is able to segment a wide variety of cells. So there were many previous softwares as well, which was used for automatic segmentation, but The older softwares were only able to do it for very specific type of cells. For example, only say some softwares were just trained purely on epithelial cells. Some softwares were just trained purely on immunostaining images. Some softwares were trained purely on bright field images. So the older softwares would work only when you have images of a very specific type. So that is the major selling point of Cellpose, is that it works on a wide variety of images because it has been trained on a very large data set with many different type of images. And then you can segment it with multiple channels. Like this is especially useful when you have immunostained images. And yeah, about these other 3 points, I'll show you in the demo. So one of the easiest ways to use Cellpose is just to use it on the website. So if you're an absolute beginner and you don't need Cellpose routinely, you just need it for a couple of images or for a couple of analysis, then you can just do it on the website. Let me show you. I'll just share my whole screen. It's very easy. This is the easiest way to use Cellpose, but it can only be used for small images. Um, Let me share my screen. So it can be only used for small images. Otherwise, Cellpose will automatically reduce the size of the image and compress it. So if you just Google Cellpose, you will get the Cellpose website. You can go there. And so they have already given some example images which you can use. If not, you can also just upload your own image as well. So I'm just going with an example image that they already have. So one more thing is that, so yeah, so for this image, it has done it quite fast. Now we can also try for, okay, I'll just explain the outputs. I forgot to do that. Yeah, so this is the original image. And after segmentation, you can see that it has drawn a boundary for each of the cell. So if you are interested in calculating the cell area, cell perimeter, or the intensity of the green channel in the image and so on, this will be a very useful thing to do. And then it also, so this is the outlines, and apart from that, it also gives you a mask image. So again, a mask is just, so if this is one cell, then in the mask output, all the pixels corresponding to that cell will have the same pixel value. So this mask output, if you are writing any codes to analyze your data after segmentation, then this mask output is what you will be using majorly. So now we can also try with any image that we can upload our own images as well. So these are the images that we'll be using for segmentation in today's class and in the Cellpose tutorial as well. So I will upload it. I don't know if, yeah, I'm trying to upload it, but it's a big file, so it's a bit slow. Anyways, before the tutorial, I'll try to be done with this, so you can also use it. Or if the upload is done by today, then you can also just um, follow along with me in today's class itself. I'll try to upload. Let's see. Okay, so coming back. Um, okay, so let's try this image. This is one of the example images. So this has uploaded. All these are very small file size images, which I am using to upload here. So depending on the size of the image and the number of cells, etc., the runtime or the time taken for Cellpose to run will be higher if it is also higher. So, and here also they are saying that if it's a very large image, then Cellpose will automatically uh, compress the image into smaller dimensions. So running it on the website may not give you good outputs. It's better to install Cellpose locally. So if you people can also parallelly try it on the website. Okay, so this has uploaded. I'll just share the link for this on the chat. So those of you who want, you can also follow along with me today's in this lecture itself. Otherwise, anyways, we will also have a separate tutorial session for this. So for that also, we will be using the same images. So if you're not able to do it today, you can also do it later in the tutorial session. So this is the link for the sample images. And yeah, this is taking a while. And I'll also just put the website link on the chat. Okay, so now it has taken a while, but it is done. So again, you can see that it has managed to get the outlines of each cell. And so if you look at this raw image, um, even by eye, it is somewhat confusing which is the boundary of which cell, because it looks like it is stained for actin and there are a lot of stress fibers, but there is no dye which specifically stains the cell membrane. It looks like it's just actin staining. But still, Cellpose has managed to get the cell outlines because it uses both the green channel and the blue channel for segmentation. So now imagine the same image without the blue channel. It would be very difficult to get the cell outlines without the DAPI channel. But since it is using both the channels, it's able to get the cell outlines and then the cell masks. So this is about how to do it on the website. It's the easiest way to run Cellpose. Otherwise, you have other options as well. Um, so other option is to install Cellpose on your own laptop. So this we will do it in the tutorial session. I think my internet itself is slow. That's why everything is slow. Um, so in order to install Cellpose, you will need a lot of other packages as well. Only if you have all those packages, Cellpose will be able to run on your laptop. So those who have, it's better to install Anaconda or Miniconda. So this is, yeah. So this one. I'll send you all of these links maybe sometime this week so that before the tutorial you can install everything. This will take some time to install. I already have everything installed on my laptop, so I can just show you today on my laptop. But the exact installation and everything, we can do it properly in the tutorial session. So the first thing is it needs a lot of packages for it to run properly. And so in order to install all these packages, you need to first install Anaconda and create a Cellpose environment. And in this environment, you can install all the necessary packages. And then once all this is done, then you can install Cellpose on your laptop. Yeah, my internet is slow, so I'll just move forward. Mm, okay, my slides are still shared. Yeah, and once you install Cellpose, it's not like, so usually whatever applications you have installed, you can just go to applications like, for example, Fiji, you can open from here. But Cellpose, you have to write some code to open it. So if you have a MacBook, you can open the terminal. So I just pressed command space and typed terminal to open it. So the first thing that told you is that Cellpose is installed inside a specific Anaconda environment. So first you have to go into that environment. So the command for that is conda activate, followed by the name of the environment, which is, so I have named it as Cellpose on my laptop. Most likely when you install also, you can name it as Cellpose. So this command will open the environment or activate that environment. Let me see. So it's saying the name of the environment is not Cellpose. Maybe I used a capital C or something. Okay, so I have used Cellpose DEV. That is the name. So once you have opened the Cellpose, then here it will come in the bracket to, then it means that you are within that environment now. So only after you are inside that environment, Cellpose will be able to open. It takes a while, like not a lot of time, but a few seconds for it to open. Yeah, so this is how the Cellpose GUI looks like. So now this is how you open Cellpose once you have installed it. So you just have to remember the name of the environment where you have installed Cellpose. So now we have opened it. Yeah. So once you open Cellpose, how do you put an image into it is that you can just drag and drop the image. So let's use the same image, image 9. So if I just drag and drop it, it opens on Cellpose. So once you open, the image is opened, you have to set the parameters of segmentation here. So these are just the display parameters, like the same histograms and the slider limits that we saw. But yeah, this is just to change the display settings. And then this will be needed after the output is created. If Cellpose has detected any wrong cells, if it has made any mistakes, then using these options, you can also correct the output of Cellpose. Then this is the main part where you will set the parameters that Cellpose has to use to segment the image. So one parameter is this diameter. So either you can set the diameter manually. So I'll show you how to do that. Or you can ask Cellpose to detect the diameter on its own. If you press calibrate, then Cellpose will automatically estimate the diameter of the cells. And whatever Cellpose is doing here, you can see in the terminal. So it seems like there is a problem in my Cellpose, but let's see. I'll just continue running it and see if it works. So the channel to segment will be the channel with which you want to get the cell boundaries. So here it is the green channel, and this channel to optional will be like a helper channel which helps Cellpose, and that would be the nucleus or the blue channel. Now if you do, yeah, sorry, my Cellpose is not working. Mm, so if there are any errors, then it comes on this terminal here. Uh, I think I updated my software recently. So one of the problems with Cellpose is that, so I mentioned that it needs a lot of um, other packages to run. So if Cellpose is using, say, NumPy 11, but then because of some updates, then NumPy is updated to 12 or 13, then it creates some problems while running it. Yeah, so it seems to be some problem like that here. Anyways, I'll try to figure this out before the tutorial session. But yeah, I can just explain the parameters by showing you the GUI. So this segmentation is where you enter all the properties. Um, so if in case Cellpose is making mistakes while estimating the diameter on its own, you can also get a rough estimate on Fiji and type it here directly. So just open the image in Fiji, and you can use the measure function, which we discussed in the last class. I'll just open the image again. It's an RGB image, that's why it's not opening properly here. So just draw a line along the long axis of the cell, and if you press M, it will the line length is the diameter. So you can do it for 4-5 cells, and then take the average, and that average value you can enter in the Cellpose diameter input. So now you have, if you take an average of all these, it would be somewhere around 50, and that you can enter here. Yeah, it's still running giving the same error. Mm, I'll just try to close and open Cellpose, because till last week it was working for me. Um, so if I'm already just I'm not within the Cellpose environment, I'm just outside on my laptop. So if I type Cellpose, it says that the command is not found. So first you have to go into the Cellpose environment and then open it. I'll just try one more time, but if it works or not. Yeah, again, it's showing the errors. I'll try to fix it by next week, or else maybe next week I can also install uninstall this Cellpose that I have and reinstall it again. That way you can also see how to install Cellpose. Yeah. So these are the important parameters, and there is one more called image pre-processing. So essentially, this will allow you to change, like, do some image processing so that the image is better to, it's easier to segment the image. It is there in the very bottom part of it. Yeah. So here you can, there are different settings that you can use to pre-process the image and then segment it. For example, if you do upsample or sharpen, then if the image is not clear and the boundaries are very blurred, when you sharpen it, it will increase the boundary clarity a bit more. Yeah, so the first Cellpose parameter is diameter. It's basically the size of your, or the diameter of the cells, and whatever Cellpose is using is always in pixels. So make sure to use pixel units. If you use micron or other units, it will not work. And then if you wanted to estimate the diameter on its own, so in the GUI, you have to press calibrate here. Otherwise, there is an option to run Cellpose with code. In that case, you have to set it to 0. If you write diameter is equal to 0, then in the code, Cellpose will automatically detect the diameter. And if all the cells have a similar size and if the image is good, then you can manually specify the diameter. But when so if also one more thing is that it could be possible that you have cells and debris, maybe some debris while you are imaging. And in this case, if you ask Cellpose to detect the diameter, it might make a mistake and detect the diameter of the debris. And then when you run Cellpose with the diameter of the debris, then it will only segment the debris particles. It won't segment the cells. So in these cases, it is better to manually specify the Cellpose, the diameter without using Cellpose. And one more thing is when you are running Cellpose on a code, sorry, and if say you have to run it on 10 images, or let's make it some 50 images, and in each of the image has different cell types or different cell sizes. But in the code, if you specify one specific diameter in this case, then it may work for the first two images, but it may not work for the other images. So in these cases, it is better to ask Cellpose to estimate the diameter. So it's based on the specific case that you are analyzing. You have to determine whether you have to estimate the diameter manually or if it is better to ask Cellpose to do it automatically. And if you use a wrong diameter, then Cellpose will definitely make mistakes in the segmentation. So this is an important parameter to keep in mind when you run Cellpose. And the second parameter is flow threshold. So it controls how strictly Cellpose uh, gets the mask output. So what is flow? So basically, Cellpose uses flow is one of how Cellpose works. So Cellpose, after you input an image, it generates something called cell flows. So what it means is that in cell flow, every pixel is assigned a direction which points to the center of the cell. And this it does by using its own CNN model. So it uses a algorithm, UNet architecture. But these technical details are not really essential. So once it gets the cell flows, from the cell flows output, it then generates the cell masks or the label outputs. So flow threshold will tell you what is the threshold flow value above which it classifies it as a cell. So the lower the threshold value, it will like it's basically less strict with the segmentation. So it will be able to detect more cells. And if you set a higher threshold cutoff, then the segmentation will identify lesser cells. But then again, if the value is too low, then it may also make mistakes. On the other hand, if the value is too high, it may miss a lot of cells. So based on the images that you use, you have to change these parameters a little bit. So the default value is 0.4. For most of the images, it's fine, but when you have cells that are slightly out of focus, then if you decrease the flow threshold, then it will be able to identify more cells. It will be nice if I can show it properly here, but unfortunately it's not working. Mm, okay, I'll figure it out later. Let's finish the class today. I think this, sorry, if you are seeing it in the phone, I think this font size may not be visible. So I'm just increasing it. Yeah. Yeah. And so one way to um, tweak Cellpose, if you're getting too many wrong segmentation, or if Cellpose is not able to detect enough cells is flow threshold, one more parameter that you can change is also the cell probability threshold. So once Cellpose gets the cell flows, it will also assign a cell probability for each of the mask. And then again, you can set a value for the cell prob threshold here. So it's basically the probability that this particle is an object, something like that. And again here, if you do a lower value, it will detect more cells. And if you use a higher value, it will be very strict in the segmentation. It won't make mistakes, but on the other hand, it might miss cells basically. And then one more thing that you have to choose is which model you want Cellpose to use. So these are the models that is already trained by Cellpose. Like these are the pre-trained models essentially. And depending on the kind of image you have, you can choose from these models. So that option is, I'll just show you where it is in the GUI. Yeah, so if you go here, you can choose which of the models you want. So if you have, so Cyto, generally actually Cyto was the model which came with Cellpose 1. And then they improved it more, and that is Cyto 2 model, which you can use when you have a cytoplasmic and a nuclear staining. And then the other model is nuclei. So this if you want to just segment the nuclear channel, then this is a good option. And suppose if you want to segment live imaging data with phase or bright field images, phase contrast or bright field images, then you can use this model. Let me try one more time. Yeah, also, by the way, the most recent model is Cyto 3. So first Cyto came and then Cyto 2, and now we have Cellpose 3 and along with Cyto 3. This Cyto 3 is the default model, but if you want to run any other model, then you have to run it from here. So I'll just try to see if maybe the Cyto 3 model is not working, if the other models work. Let's see. I'll try Cyto 2. So I changed the model, and then here in the terminal, you can see that it is loading this model. So right now, whatever you do in the GUI, you can see what is happening in the backend in the terminal. So instead of doing it in the GUI, you can also write the code to do all of these, which is slightly more advanced. If you're comfortable with coding, you can do that. Otherwise, you can just use it in the GUI. It's running. Yeah, again for Cyto 2 model, it is showing the same error message. But it ran so for this model that I tried first, it actually ran, but it could not detect any of the cells. Maybe let's see, I'll try again. So if you see the outputs here, so when it is running, it will just buffer like this. Just wait for a few minutes. Okay, when this is not running. And one more thing is that you can run Cellpose on CPU or GPU. When you run it on GPU, it will be faster, but to use Cellpose on GPU, your computer has to meet some requirements. If the requirements are not met, then it won't run. Maybe I'll just try to run it without the GPU if it works. For example, if you have a Windows, then there are specific NVIDIA cards, GPU cards specifications, only above that will work. It seems to be working in the CPU without the GPU, so maybe that was the problem. Let's see. So in this image, you can see that most of them are bright, but there is one very faint cell here. Let's see if Cellpose is able to detect this or not. Like as I said, in CPU, it takes slightly longer to run. So on Cellpose is better to run in Windows, I would say. Yeah. So now it has detected, but it has made a lot of mistakes because we did not calibrate the diameter. So it has detected very small particles. So now let's try to calibrate the diameter first and then run it again. Yeah, so I was saying that Cellpose is mainly written for Windows. So things like running Cellpose on GPU, training a new model, these are all better on Windows than on Mac. So essentially, they first wrote it on Windows and then it was adapted to Mac. So some features that are available on Cellpose Windows is not available on Cellpose Mac. I'll come to it in the upcoming slides. It also runs on Mac, just that a few features are not available. Like for example, in my Mac, it's not run. My Mac has GPU and it is compatible with Cellpose, but still it is showing some errors. So with CPU, even just calibrating the image is taking time. I'll just try to close other programs so that Cellpose has more space to run. Yeah, I've closed everything else. Yeah, so now it has estimated the diameter as 50. So, but this is a very large cell compared to the others. So let's see if Cellpose detects just the small cells or if it can also detect this big one. I'll run this. Yeah, like I said, because this is a big cell, it has not detected this. Instead, it has detected some part of the cell. So these are actin cells that have been stained for actin, and this is the actin lamellipodia in front, and this is the cell body. If you want to check if I, so it has just detected a part of the lamellipodia as a cell, and it has also not detected this faint cell over here. And this one is oversaturated. Yeah. So this is, it has correctly detected these two cells as two separate cells. And here, like you can change how you want to see the output. You can see it as an outline image or you can see it as a mask image. So now I'll try to decrease the flow threshold because it has detected a wrong cell here. So you have to go to additional settings. Here you will get the options for flow and cell prop. So the default are 0.4 and 0. I'll make it half and see if now it gets rid of the wrong segmentation. Mm, yeah. So now you can see that it has not detected it, but it has missed other cells as well. So this parameter you need to play around a little bit. Yeah, so now it is better, like it has detected. Okay, it's still going on. Yeah, so this point 0.3 value has worked better. It has detected like the small cells and it has not made the mistake here. And for this, I used Cyto 3 model. Now, for example, if I try to run this with the live cell model, which has been trained for, or I'll try to use the nuclei model, then the it will segmentation may not be very good. Yeah, so it has not detected much because none of these are looking like typical DAPI stained nuclei. So you also have to be careful about which channel, which model you use. And if you see that it is not detecting a few cells, or if it is detecting too many cells, then just play around with the parameters a little bit until it gets the segmentation right. And I was also telling you about some differences between Windows and Mac. So the main thing in Mac is that Cellpose 3 they are still developing for MacBook to have all the features that are there in the Windows version. So one main feature that is missing is that so in Mac you can download like either Cellpose version without GPU or with GPU. So looks like the one that, so mostly I generally use the Windows Cellpose that is in our lab, but sometimes I use the, like very rarely I use it on my Mac. And this is the version that I have. Sorry, Cellpose. Yeah, the Cellpose version without GPU, like I said, and we also saw in these images, it's quite slow. But the Cellpose version with GPU on Mac, it has the problem that the GUI, some features of the GUI don't work in the GPU version of Mac. So the main feature that does not work is the uh, option to draw and remove masks of the output. So if Cellpose has made any mistake and if you want to manually correct it, then unfortunately that option is still not there in the Cellpose GPU, Cellpose version, which has GPU on Mac. That's why now I remember I had downloaded the version without GPU. That's why the first time I was trying to run Cellpose with, uh, I had ticked use GPU, but it was showing the error. That was the reason. Um, So once what so Cellpose is like an open source software. It doesn't need any subscription or payment, etc. You can just download it, and it is continuously under improvement. So these if you want to get the latest updates about Cellpose, you can go to the GitHub page of Cellpose. So here people will post about what is the latest version they have, what are the changes that they have done from previous versions, and what are the errors that people get when running Cellpose. And then the people who have developed Cellpose will also tell people about what are the updates that they have done to get rid of the errors. For example, in Feb 2025, they have made these changes. Yeah, so this is what I have saying that and this is actually specifically for the silicon chip Macs, like the M1, M2, M3, and so on. So because this is so it's not all the Macs, it's the new Macs with the M1, M2, basically the silicon chips. So the problem that they say is that for the silicon, so basically Cellpose needs a lot of ML packages to run, and these ML packages that Cellpose itself uses have not been written for the or they are not optimal for the silicon chips. So that's why some of the features which work on other Macs and which work on Windows does not work on the silicon chip Mac. And here they are saying that so they have corrected some of these issues, but they are still working on the ability to draw new masks with GPU on Mac. And here there are also other options like how to install it and so on. And suppose when you run Cellpose, if you get any error or if there is something that you want to tell the developers to include as a new feature, then you can open an issue. Yeah, so if you go here on GitHub, these are if you and then you can just open a new issue and say that you saw this bug or this is a new feature that you want in Cellpose or you're not able to install it and so on. And you can, so suppose you're getting an error message, you can also check in the issues here. If somebody has already opened this issue, then you can just, so like somebody has reported a bug here, and then the developers reply to it. And if somebody had already reported any error that you get, then you can just see what they have replied and implement the solution from there. So the Cellpose GitHub page is where actively it's still being improved because it's an open source software. And one more thing is whenever you use Cellpose, you have to cite the Cellpose papers. So because it's an open source software, the developers are not monetarily compensated for it. So one way that you can acknowledge them is by referencing their paper, so then they get more citations. And yeah, these are the options that you have to give in Cellpose for image pre-processing. So this is the raw image. I'll try to upsample it. So this doesn't take a lot of time. And then again, calibrate it because the diameter is not correct. So for some of the images, after doing a image pre-processing, it works better. But if the image is already good, then pre-processing won't make much of a difference. So these are what you have to cite. Okay, it's still estimating the parameters. Now I'm running it with the 0.3 flow threshold so that it can run better. But in this case, the image itself is quite clear. So it, like, the previous version itself gave good segmentation. So pre-processing has not made much of an improvement because the previous version itself was good enough. So, and also I was telling you about the, okay, I'll come back to it later. This we did finished. Um, yeah, so this is like, once the Cellpose has segmented, you can also change or correct the segmentation if it has make mistakes. So it has not detected the cell, so you can just, so it's, you can just draw an outline around it. So for Windows, if you're using a normal mouse, or even for Mac, if you're using a normal mouse, you have to right click and then it will show you that red circle. And then you can just move your mouse to draw the outline. And when you come back to the red circle, the mask will finish and it will be created. And also this one. Yeah, so now it has created these two masks. And now you can save the output. So, like, there are different, it's the same output, but there are different formats. And the outputs will be saved in the same place where the input image is there. I'm just saving all the forms of output. Okay. So this is about how to add a mask. I'll also show you how to delete wrong segmentation. Now I'm setting a very low value of low and the cell prop threshold. Now if I run it, you will see that it makes a lot of mistakes and detects extra cells. So in those cases, you can manually remove the outputs, remove the wrong masks. Yeah, so it has made a lot of mistakes here. So one way to remove mask is you can just go to this part, and if you do click select, then the mask that you select will be removed. But in this case, you can see that all the mistakes it has made are in this specific region. So you can just do region select and move this box. So whatever masks are within this box will be deleted. And then you can draw the correct outline for this. Yeah. So this is about how to manually correct the segmentation outputs. After that, yeah, to draw the mask, you just have to right click and then a red circle will come. And then just drag your mouse along the cell outline. Yeah, and this is how you correct the Cellpose output. There are also some video tutorials on the Cellpose website. So actually, this is the first version of Cellpose V1. You can see that the GUI is quite different at that time compared to how it looks now. So it's not just the GUI, but the models are also better. So now let's talk about the different types of Cellpose outputs. So one main output is called the label or the mask output. So in this type of an output, it gives you a file such that whatever um, the same cell has the same pixel or the value assigned in a matrix. So here all the green objects have like 3 assigned to it in a matrix form, and then whatever is in the building is assigned as 5, and all the roads are assigned as 4, and so on. So if you open a label or the mask output, this is what it represents. So I'll try to open this label output in Fiji and show you how it looks like. And yeah, you can see that whatever you do on the GUI, it also comes on the terminal here. So these are the Cellpose outputs. Let me, that I just saved now. So this is the mask output. Let me open it on Fiji. If you just open it, normally it will be a black image because the pixel values are like 1, 2, 3 for each of the cells. So you have to open it in Fiji to visualize it properly. Mm, yeah, so you can see that all. Sorry. This one. Yeah, so this is the histogram and the values are from 0 to 13. Like all the pixel values of 1 will correspond to one cell. And if this is one cell, then it all of these pixels have the same value. And you can see that even in the image histogram, it's very discrete. It's And then if I oversaturate it, then essentially now, so earlier this is like an instant segmentation because it has differentiated the cells from the background, and it has also differentiated between the cells. But now if I oversaturate it, then all the cells get like a value of 1, so it becomes a grayscale image essentially, where all the 0 pixels are stained for 0 pixels represent the background and all the 1 pixels represent the cells. So then it what was the other type? Yeah, so then it sort of resembles a semantic segmentation in this case. um, yeah, so this is about the Cellpose mask output. Apart from that, you also have the NPY .npy format. So this .npy format, this you can, like if you want to open it in Python code and do more processing, then this will be the best option. And this option you can also reopen in Cellpose again. Sorry, it keeps changing this. Yeah, this you can reopen in Cellpose again, and it will have the input, output, what are the parameters that we used. Everything will be saved in this format. So mostly this format is used for um, further analysis. But this format is used if you want to save the segmentation analysis itself with the parameters that were used. So generally, if you are, say, going to publish something in a paper and you have used this kind of Cellpose analysis, then it's good to have the .numpy file so that you know exactly which parameters you used. And if you want to make any corrections or clarifications, then you can go back to this file and check what you did. Not just for image analysis, but for any kind of analysis that you are doing for any publication, it's better to have the analysis parameters as well. So this is, all of this was about how to run Cellpose on the GUI. It's very, like, it's again an easy way to run it. But suppose you have a lot of images, maybe you have some, I don't know, time lapse data, like let me see if I have something, because whatever I used are just like some sample images, but in the real world, the data is much more complicated and you also have a lot more data. Yeah, so let's take this experiment that I have. So then I have like multiple images for just one output, like one scene where the cells are moving over time. Now, if you try to do, so this is just like one scene from one experiment. And like this, I have 10 per experiment or 15 per experiment. My laptop is, yeah. And now if you want to like do each of these manually, it will waste a lot of your time. So then in that case, it's better to use Cellpose on a code. That the code that has to be used is already there in the uh, Cellpose website itself. You can either download it and run the code. It's a Python code, so if you are able to run Python on your laptop, it should run. But if you don't have Python on your laptop, or if you're not comfortable with coding on Python, then you can also run it as a Google Colab notebook. So let me see. Suppose actually there is a notebook which opens directly on Google Colab. But the problem with running it on Google Colab is that I think there is a, in the free version of Colab, there is a specific limit on GPU usage. So if you are segmenting for too many images, it may not work for all the images. Yeah. So one reason why a lot of people use Google Colab to run codes is that it has GPU access. So even if your laptop does not have GPU, then you can go for Google Colab. But yeah, but you can't do it for a lot of images because the free version has a limitation. But if you subscribe for it, I think you can use it more. One more thing is that to upload images to Google Colab, it has to be uploaded on Google Drive, and then you have to give access for the collab to your Google Drive. It's not really like a big deal, but still, this can be done easily, but it's just a hassle, right? Like if you have some GBs of data, but I think um, Google Drive, the entire storage in the free version is only 15 GB, but there are some experiments in which one experiment itself you can generate 50 GB, 100 GB of data. So again, that will be a problem if you just have the free version. Again, you can pay for it and get more storage, more GPU usage, and so on. Uh, block. Yeah, so this is the Google Colab notebook. And here you can change your runtime. So this will tell you, assign you to from which server this code will run. So once this is done, you can start running the code. So just so in this Google Colab notebook, they have already written the codes, and you just have to like run all of the cells. So first you have to install Cellpose. And if you want to make any changes to this, then you can create a copy, save a copy to your drive, and then make the changes in that. This will take a while. Yeah, and this is the part where you have to give access to uh, Google Colab to access your Google Drive. And here you have to set the parameter. So this is in case you want to train your model, but it's not needed if you don't want to train it, if you want to use a pre-trained model. And this is if you want to use a pre-trained model. Yeah, you don't have to run all the cells when you run this. So this, they say it comes normally, it's fine. Now we have run the first cell. And now this was what I was saying, that Cellpose needs a lot of other libraries to run. So all of these other libraries you have to import and do all of that. Mm, let's see. So it's not able to import the models. because the first cell itself did not run properly. Mm, so again, this is one common thing error that you get in Cellpose is that the versions don't match. Like for example, it says that it requires lesser than this version, but in my case, I have a more updated version, so it's throwing an error message. This is weird because the code asks us to install this version, but it says that it needs a previous version, so I'm not sure what's going on. Let's see. Okay, now it seems to have worked. Now let me. Hmm. Hmm. So I'm using this GPU version here. Now let me run it again. No. Maybe try this. If Google Colab is giving problems, you can also run it on Jupyter Notebook, or any other um, Python compiler. So a lot of people have already made these Python codes that you can run. I think it's there in the Cellpose website itself. Let's see. Okay, so they have some Google Colab notebooks on their website itself. Let's try to, maybe this would work better. This keeps. Yeah, and then it will ask you to sign in when you connect your Google Colab to your Google Drive. That's fine. Mm, you need to give these permissions. This is a different. So, I was already like previously I had used this for some image analysis, so I already have some images here. Uh, okay, maybe we can use the segmentation folder that I had uploaded here. Lectures, tutorials. Mm, yeah, so we can use one of this images, uh, and then copy the path to it. So first I'll install Cellpose. So here the good thing about these Google Colab notebooks is that they have already written the codes and they also have instructions on how to run it. So it's easier than running it on your own laptop. So if you're not able to install Cellpose on your laptop or if it is too slow or so on, you can just run it on Google Colab. And this is just using importing all the needed libraries. So here I had copied the path to this image and I am pasting it here. Okay, so here it is saying it has to be a directory. So it will run for a directory, meaning folder of images. So I will just give this entire folder path and see if it is able to run for all the images in that folder. And yeah, the path has to be within quotes. So it has said that it has found these three images. Yeah, it's only so I have some png as well, but um, it's only identifying .diff because of the code here. So let's see now if it can identify. Hmm, still identifying only diff. Okay, I'll try to figure it out later. So here it will say for each image, it has how many channels. So it has, okay, so this image is actually a Z-stack image. And the other TIFF files are just a single image file. It does not have any Z-stacks. So that's why it is saying that it has identified these many 15 planes. Just give me a minute. I'll be back. Hmm. And here you need to set the parameters like, so in the GUI we were setting it here, which is the first channel, which is the second channel. In the code, we have to type it. It's the same thing, but we have to type it. I think these, let me just see which channels to use for the images, and then we can type in the code here. This is the, so this is also a big file because it has these tags. I think this is just a face contrast image. So for a face contrast image, the channel, oh, okay, these are just, it's not face contrast, but it's just a single gray channel image. So for gray, the code is 0 and it has just one channel. So the channel 2 is none. And we can type 0 and none. And so, yeah, this is the actual code. But in this Google Colab notebook, they have made it easier for the user with these kind of buttons. And here you can also change the flow threshold. Flow threshold, we saw that 0.3 had worked better. So now it's running the segmentation. Okay, so it is giving an error because we have this TIFF file, uh, this one, which has 5 Z stacks and 3 channels. So if this is the case, then the software gets confused which image to use, which channel, which Z stack image to use and segment. So I will just make it into one single TIFF file by doing Z project. And here you can see that from the first Z plane is out of focus. The second Z plane has the basal actin stress fibers, and then the top planes have the cortical actin information. So because all the Z stacks have some information, it is better to do a average intensity projection. And the first Z stack is out of focus, so that is not needed. So I will first make a Z projection to make it into a single image. Otherwise, the software is getting confused. Z project. And I will start at the second slice because the first slice is out of focus. Huh. So now it is like a 3-channel image, and I have saved it here. And I'll also upload it to the Google Drive and try to run the code again. So here I'll remove this. So for whenever you're using 2D Cellpose, make sure that the input is not a Z-stack image. If you give a Z-stack image input, it will just throw an error and the code won't run. This is the one, right? This one, yeah. Yeah, so it's still uploading. So if you you can also try to run it parallelly with me. Where is it? This one, right? Okay, so I'm sharing this link. You can make a copy of it and run it on your Google Colab. And I already sent the link to the images here. No. Yeah, this is the folder with the example images. So now hopefully it should be able to run. Let's see. Okay, I'll try giving the image path again because I think it has already read the images in this code. So maybe I'll rerun from these. Yeah, now it has detected the Z-stack image. Now it has taken 3 as the size. So for the first image, we can try to segment with the red as the primary channel because the red channel has better cell boundaries, and blue can be the secondary channel. So the primary channel is 3, red is 1, primary channel is 1, secondary channel is blue, and it is asking for one more channel which can be green. So red, RGB, and green is 2. Let's see. Here, flow threshold, batch size. What is batch size? I am not sure what this argument is. But let's see. Because this is not there in the Cellpose GUI. Hmm. Mm, um, Maybe I think it's not running properly for the RGB image, like the multi-channel image. What is it saying? I think the problem could be the diameter. One channel, please let's see now. Okay. So it is compressing the file size from 16-bit to 8-bit. Why is it doing that? Anyways, we'll try with the other phase contrast image. So maybe this one I will move it. So let's see whether it is able to segment these two. I generally use Cellpose with my Jupyter notebook. I have not really tried to run it on Google Colab. So, okay, so now it has identified the image properly. This is a face contrast image. So it's just 0, comma 0. Both are gray channel. Now let's see if it works. 2 indices of our array. It's a single channel image. That's why, but I selected 2 channels here. That's why it's giving an error. It should work now, I think. It is still saying the same. Now it will just say no channel found. But, so this code will or maybe it's not working because this code is written for just one image, but there are 2 images in the folder. So I'm running this code block, which is written for a folder of images. So it has processed, it is processing the first image. It also has to process the second image. So here, this code block will run the segmentation. Actually, I think this we could have given it as 0. It's fine. And then in this code block, it will display the outputs. Yeah, so it has run and the outputs are saved here. Yeah, so I think that was the problem then. Previously, I was running a code which was written for a single image, but my folder has many images. So if you have a folder of images, run this code block. So here in this line, it is first reading the images. Oh, okay, I'm sorry. I think both of them are for running the images, running the code for all the images. But it's just that here you have to use it if you have many large images. But if you have just small images, then you can use this block. And yeah, the outputs are saved here. Let me download this and check what it has done. But yeah, it should be in downloads. This is the output. And which is the original image. This is SA-T29, this one. Yeah, so it has done the segmentation properly. So in this folder, I just had 2 images, but if you had many images, then if you just run the code and leave it to run, then it will finish it for all the images. So similarly, it has also given the mask output for the next image. So now let's see, once you have these outputs, what can you do with these outputs? So now we have done the segmentation and everything, but we want to analyze it more. So for that, there is this plugin. Again, this is also open source, so you have to cite it when you use it. And first you have, this is not a pre-installed plugin on Fiji. You have to separately install it on your laptop. So if you go to this website, it will have information on how to install this plugin. I can close everything else. So yeah, this is how you run Cellpose on Google Colab. Why is it? I don't know if my internet, like ISC Wi-Fi is slow. It is fine. So this is the name of the plugin called LabelsToROIs. So this is the person who has created it, and he has made it open source for everyone to use. But somehow this page is not working now. Okay, yeah, so it's working now. So to download this plugin, you have to download this file and then move it into the plugins folder. I think this I covered in the second lecture, I think. Yeah, second lecture. So in my Fiji, it is already installed, so I'm not going to do it now. You can try to do this. If you have any doubts, you can ask now, or you can ask in the tutorial session as well. So so to use this plugin, you have to open, actually you don't even need to open these images on Fiji, just it will ask for the file path. There you have to give the file path, LabelsToROIs, and now let's say we are just doing it for a single image. So you have to give the path to the original image. It's in desktop segmentation, and this one S8. This is the one that we segmented on Google Colab, and the mask image S8. This is the S10. S10. Okay, the mask image I downloaded it from Google Colab, so it's in the downloads folder. How do I? Yeah, this one. Now just do next. So what it has done is it has converted this mask output into a ROI output. So once you have the ROI output, then in the last class we saw how to measure the cell properties from the ROI output. So you can go to set measurements here and select what are the parameters you want Fiji to output for you. So area will give you the area of the cell. Center of mass will give you the centroid coordinates of each cell. Both will have the same values more or less. Some decimal point difference will be there. And then perimeter will tell you the perimeter of this ROI. Mean gray value will tell you the pixel intensity within each outline. So, for example, if this outline has maybe 5 pixels, it has more. I'm just giving an example. If it has 5 pixels of values, I don't know, 100, 200, 300, 400, 500, then it will give you the mean of all these five values, which in this case is 300. And then if you do fit ellipse, it will fit an ellipse to the size of the outline of the cell and give you the value of the major axis and the minor axis. And similarly, median will give you the median value of all the pixel intensities. Again, mean, median, mode, these are just the typical statistical outputs. Um, yeah, and stack position is not needed here. It's just a single channel image. And you can select how many decimal points you want. The default is 3, which works fine. And then if you do save CSV table, then you can see that it gets saved in the folder where you have the original image, and it has the output for all the 5 cells. So this is easy. It's just 5 cells, but you can imagine a scenario when there are many more cells, you will have like many more outputs to work with. And then this CSV file, then you can use to do other kinds of analysis. For example, you might want to check if cells of smaller area have higher protein intensity, or whether it is cells with less area that have higher protein intensity, and so on. So yeah. And again, this was for a single image. But if you have a folder of images, then you don't have to manually click and do it for each image. So for a folder of images, I think in this itself they say, if you go to this website, it's pretty detailed explanation of how to use it, but I can also show you now. And yeah, you can also watch this YouTube video where he has a demo on how to use it. If you want to use it for multiple images, the mask images have to be labeled in a certain way. Yeah, so the output image has to be labeled, it has to end with underscore label.png. So if the name of the original image is this one, SAT29C2, then the name of the mask image has to be the prefix of the original image and the suffix underscore label. So rename underscore. So this is how the Cellpose output is from the GUI is saved with this suffix underscore CP underscore masks. But for this plugin, it has to be in this format based on the code that they have written. And similarly for this one, I will also, and the original image and the label image have to be within the same folder. So, but the other label image goes in a different folder. So I'll also put that in this folder. But where is it? S8 mask. And this is the raw image. Okay, yeah. So now, again, it's the suffix is underscore masks, but it has to be underscore label. Only then the plugin will be able to recognize that this is the original image and this is the mask image. So name of the image prefix and underscore label suffix. Similarly, name of the image is the prefix and underscore label is the suffix. And if you want, we can also have I okay, run it for these, run Cellpose for these two, and then run labels to ROIs for 4 images in one go. Sorry. Uh, mm, Now I'm unticking use GPU because that was not running properly. Now I have to set the channels as well. Primary channel is the green channel, secondary is the blue channel, and then run. So now it has, yeah, so this segmentation is good enough. So I'll save this output. Right now we are using labels to ROI, so I'm just saving the masks output. But it is good practice to save the numpy output as well, the .npy output. So again, this is a different image and the cell sizes are different. So you have to calibrate the image again and then run Cellpose. So the sizes have changed. So now let's say that I give a very small diameter size, and if I run it, it won't be able to detect cells properly. So it has uh, terminal, yeah, it's still running. And I hope you can still see my screen and everything. Yeah, it's still being shared. Okay, great. So for smaller diameter, it takes longer to run, which is why it's taking time. Yeah, so because we set a smaller diameter, it has detected like some parts within the cell which is not correct. So yeah, it's very important to set the diameter properly. Okay, it's done. So now so I'm not going to save this output because it's all wrong. So I'll calibrate and run again and save the correct output. So it has calibrated. Now I'll run it. Yeah, so for a bigger time meter, you can see that the runtime is also slower. I'll also save this, save masks. Now, before we can run the labels to ROI plugin for a folder of images, we just have to make sure that the file names are in the correct format. So it has to end with underscore label. Rename. Okay. So now let's run the plugin. Actually, it's been 2 hours since the class started. So we can take a 10-minute break now and be back at 12:10, and then we can continue the lecture. Okay, so let's continue where we left off. Um, so we were segmenting all the images in this segmentation folder, and we have also renamed the Cellpose mask output images as per the requirement of the Labels to ROIs plugin. So now let's run it and see if it works. Fiji is here. So go to plugins and Labels to ROIs and select multiple images. And now you have to give the folder path where all the raw images and the Cellpose output masks are there. It's in desktop and segmentation. So I'll just, yeah, so these are all the parameter outputs that you want in your CSV file. So just check once if whatever you need are selected here. Okay, and now you can run it. Yeah, so for each image, it has, yeah, here, kind. So we had 4 images, S8, S10, mm, and it has saved the CSV files separately. And if you can also open the CSV files and check whether all the outputs are there properly. So you have area, mean, mode, and these are the centroid and the center of mass coordinates. Like they are essentially the same. It's just a matter of a few decimal point difference, and then you have the perimeter. And it looks like so I'll just and it looks like the values are in pixels. That will depend on the original image. So this is no this output is for S8 file. So let me open that. Yeah. So actually, in this the calibration is wrong. It's saying that 2048 pixels is 6 inches, which is not correct. So that's why the area values are absurd. So one more thing to keep in mind is to set the scale properly before you run labels to ROIs. Otherwise, it gives outputs in the wrong units. So I'll set the scale and other properties for this image. So the conversion, this was taken in 20x. So the conversion factor is 0.325 micron per pixel. So after you run it, just open the values once and check if the values make sense physically. Like the area values that we had were uh, some 0.06, which does not make any sense. And we were also not sure what were the units because I didn't calibrate the image. So now I'm calibrating it. And yeah, this one also is calibrated wrongly. Micron, micron, and 325. So now I'll run. After calibrating it, make sure to save it. I don't know if I saved the previous one. Let me check. Yeah, I calibrated, but I didn't save it, so it is still in the wrong units. Sorry. This is micron. Micron. 325. So calibrated and Control S to save it and replace it. So now same thing. Plugins, labels to ROIs, multiple images, desktop segmentation. Now run it. Mm, yeah, so I guess these are the new outputs. Yeah, now the area values and the values of the major and minor axis make more sense. So typically, these cells are around 20 to 40 microns, and this we also get a similar range of values here. So now it seems that the values make sense. So yeah, that's pretty much what I wanted to cover in this class to show how like some basics of Cellpose and um, some basics of segmentation and how to use it, what are the different kinds of inputs that you have to give, what are the different kinds of outputs that you can get, and once you get the output, how do you further process the output using this free to use plugin called LabelsToROIs. So that's pretty much it for today. If you have any questions on this, we can take it. Otherwise, we can go to the recap quiz because we still have some time left. Mm, okay. So if there is no other question, I think a lot of people missed the previous recap quiz. So you can take until maybe 12:45 to do this, to finish the quiz and send me the answers. And then in the last 10-15 minutes, we can discuss the answers to it. So I'm putting the link to the questions in the chat box. Yeah, so this is maybe you can send it before 12:45, and then the next last 15 minutes we can discuss the answers to this. So in this, some of the questions are you need to take some time to think about it. It's slightly more advanced than the previous recap quiz. Yeah. So at 12:45 we can start discussing the answers for this. Mm. Um, I haven't got the answers yet, but um, it's about time. So let's start discussing the answers. Mm, so sorry. So the first question is, if you want to tell whether two images are identical, is comparing their histograms always a reliable method? Option A, images that look the same can contain different pixel values. Images that look different can still contain the same pixel values. Images with the same pixel histogram are the same image. Images with different pixel histogram are different. So if you look at the first option, it says that images that look the same can contain different pixel values. So generally it depending, so let's say it is possible that even though to our eye the images are the same, there could be some small differences in pixel values that we may not be able to see visually. So A is correct. Option B, images that look different can still contain the same pixel value. Yeah, so option B is also correct because we saw that the same pixel value can be represented visually in different forms depending on which lookup table you use. You can represent it with a cyan LUT, blue LUT, or you can just make it a grayscale image and so on. So option B is also correct. The second question is, which ROI type is best for measuring cell perimeter? So generally cells are not, they don't have a specific rectangular shape or an oval shape. They have more of a irregular shape. So it would be best to use a freehand selection and draw the outline so you can get the perimeter properly. The third question, what happens if you save an image as TIFF? Maybe because I have still not got the answers, I'll just discuss the MCQs today. We are also running short on time. And once I get the answers, you can take some more time with these subjective questions because they require you to think a little bit. So you don't have to do it in a rush now. I'll just discuss the first up to the first 5 MCQ today. These are more or less straightforward. Uh, 6 to 12 are you have to slightly think about it, so you can send me your answers whenever you're done, and we can discuss the solution to these in the next class. So I was at the third question. What happens if you save an image as TIFF without saving the ROI separately? So here what they're asking is, if you open an image and if you just draw the ROI, but you don't save it, and then you save it as a TIFF file, then if you don't save the ROI, it's not saved. Like the only when you save the image as TIFF, only the image gets saved. The ROI is not saved. So the ROIs, they just vanish unless you add it to the ROI manager, and then in the ROI manager there is an option to save the ROIs as a separate file. And then the fourth question is, in a merged RGB composite, what does turning off a channel in the channels tool do? It deletes the channel, it hides the channel, converts into a binary mask, reduces the bit depth. So it is a composite image. So in composite image, let's say you have red, green, and blue channel. All the 3 channels are stored separately, and depending on which of the channels are turned on, only those are visualized or they are shown on the display. The other channels are still saved in the image file, but if you turn off a channel, it's not visualized or it's not displayed on the screen, but it is still saved in the image file. So what it does is it just hides that channel visually, but the channel is still there in the image file. And then the fifth question, the status bar also shows the x and y coordinates for the pixel under the cursor. So when you, I'll have to share my entire screen for this. Okay. So this would be easier to explain with Fiji open. When you open an event, move your cursor to a particular point, then if you look at the top on the Fiji bar, then it shows you some information. So let's open this, for example. And now if I keep my cursor here, it shows the x coordinate and the y coordinate and the value, which is the pixel intensity value. So you can see in this image that if I move along the background, then the values are around 337, which is the background intensity value. And if I go within the cell, the values are in the range of about 7 minutes. Sorry, 700. Yeah, and this is a slightly lighter intensity. And it also shows you the x and y coordinates. So in this question, they are asking, where is the 0, 0 coordinate? So let's check, is it in the center? No, in the center it's around 1000, 900. Is it, so, and if you go down, you see that the y value is increasing as you go down, which means that the y coordinate of 0 is on the top. And if you go to the right, you see that here, right now x is 144, and then it increases as you go right. So which means that x0 x coordinate is on the left side of the image, and the 0y coordinate is on the top of the image. So the top left is where you have the centroid of the 0, 0 coordinates. So this question, if you had just opened and played around with Fiji, you would have got it. So it's the top left corner. Yeah, and for the rest of the questions also, you can try and use Fiji and try to answer these questions. Then it would make it slightly easier, and we can discuss these in the next class. So that's it for today. If you have any questions, we can take it. If not, we can meet next week. Okay, so I hope everything was clear today. We can meet. So next week, I'm not sure exactly when the class would be. If it's going to be on a Saturday or a Sunday, I will confirm it and send you an email if there is any change in the class timing. If I don't send an email, it means that it's going to be in the regular Saturday 10 to 1 time slot. If there is a change, I will send you an email to notify. That's it for today. Thank you. And see you next week.